but we see you.
1, 2, 3, 1, 2, 3. Ladies and gentlemen, we are live. And not only we are live, as we promised, we are back on the road. We are back, babies. I can't be more excited. I am in Bordeaux. I am in the city of wine, in the city of photonics. I am in the city that is hosting the European Conference on Optical Communications, ECOC, and here, from the booth sponsored by the Photonics 21 and the European Commission partnership, the Pilot Line booth, I am making an event together with EPIC. We are all together, the EPIC team, on the EPIC meeting on spectroscopy and process monitoring. Welcome to one more online technology meeting. On Monday, September 13th, the discussion topic is process monitoring. We're going to build on previous discussion on smart manufacturing to bring together leading companies involved with real-time inspection and analysis of the production process. Whether you're working in automotive or the pharmaceutical industry, process monitoring is essential for safety control, production optimization, and automation. Successful production companies never take this lightly. Remember back in 2015 when chemical giant BASF created its own spin-out company, Trinamics. A chemical company creating a near-infrared spectroscopy company that was shocking for many and obvious for a smart few. Since then, we have seen a significant flow of investment by production companies into novel photonic inspection technologies. And that makes me extremely excited. So let's show those smart production companies relevant trends that are happening now. But to start realistically, let's understand the demands from one of the main companies in the pharmaceutical world. For the first time at our EPIC events, we welcome the presence of end user Dr. Reddy, a $2.4 billion multinational. This is truly a unique opportunity to meet and discuss with their head of advanced characterization. What are their unmet needs? Let's make epic things happen. Perhaps another Trinamics is on the horizon. In my mind, 4 major developments are happening in this area. Number 1, Terahertz. Substrate-independent ultra-fast multilayer thickness measurement has been proven in an amazing collaboration between Optical, Fisher, and Fraunhofer HHI. They can simultaneously measure the thickness of up to 5 layers with an accuracy of less than 1 micrometer. Number 2, digital holography. Have you heard about 4D profile measurements? Yes, as Isaac Newton said, we live in a 4-dimensional world. And with digital holography, using a 3D profilometer, we can enable time-resolved measurements. And time-resolved is our 4th dimension. Number 3, hyperspectral imaging, where process cameras are getting more and more compact every day. And number 4, miniaturized spectroscopy solutions in the visible, shortwave, midwave, and longwave infrared with form factors and cost small enough that allows them to be positioned in every corner of the manufacturing line. I expect great things will happen on Monday, September 13th at 3 p.m. Do not miss this one. Sign up immediately. No excuses. Be there. And you sign up immediately with no excuses and you join the online technology meeting on spectroscopy. We are really excited what's going to happen. We have amazing companies in the room like Hydroctis, like Dr. Reddy's. Let's get the meeting started. I don't think with any further ado, we need to get the ball rolling. Elena, you have done a fantastic job over the summer bringing these fantastic companies together. EPIC is back alive, but we continue our online experience. That's not going to finish. We continue with our online technology meetings every Monday at 3 o'clock. No matter where we are in the world, we'll have our fantastic meetings. Thank you so much for the amazing support you have given us. We are a team which dedicate their life to the photonic industries. And as you very well know, we organize events, provide access to our network, help you raise capital. We have the biggest website in the world to find a job in photonics, jobsinphotonics.com. And we have great market reports, and we're making even more market reports. Look at the one that we published 2 weeks ago already for breast and prostate imaging. Quite a spectacular, great job, our head of photonic market research, Tracy Vanik. When we continue also with the season 5 for the online technology meetings, today is our number 2, but have a look at what's gonna happen next week on Monday. Laser ablation and laser cleaning. We have amazing success stories. Pay attention to what Forjet is doing. Forjet is doing on cleaning tires with lasers, a huge, huge market. And also, but today we also have to continue telling you that we support the quantum industry. Our first meeting the 6th of October, atomic clocks and network synchronization. Today we organize our online meeting on spectroscopy and imaging for process monitoring. There is no better person to take it over from here than Dr. Elena Beletkaia, who has worked extremely hard with the right companies here. But before that, Elena, you have to say that this event actually gotten the support of so many companies. It wouldn't be possible with the support first of our collaborators, EMBA, European Machine Vision Association, and also our media partner, Laser Focus World. But most important, Laser Focus World, most important, this event wouldn't be possible without the support of our sponsors today, all the way from Netherlands, Admesy. Admesy provides colorimetry solutions for any part of your process monitoring, whether you make LEDs or you make any kind of fluorescence device that means to collect fluorescence, you want to know the color of every part of that device. Admesy provides those colorimetry. Art Photonics, from the visible all the way to the mid-infrared, a specialty fiber made in Berlin. Whether you're in the medical industry or the chemical industry, you need a fiber to transmit light in any wavelength. Art Photonics, your partner. You're looking for DFB lasers, you go all the way to Ireland. And then we have Eblana Photonics, providing DFB laser solutions for any spectroscopy demand. If you're looking for any optics or any coatings or any process that you need to do with optics, Edmund Optics is your partner. You're looking for semiconductor lasers. There's a company in Finland called Modulight. Modulight provides AMBE growth from wafer all the way to device packaging. Modulight provides array solutions to even final end users doctors, the entire vertically integrated company in Epic. If you're looking for coatings, the company is Vortex. Vortex provides optical coatings, but they are very good, especially in the mid-infrared. If you need mid-infrared filters on coating, Vortex is your partner. For 20 years, we are having Ocean Optics being the partner providing spectroscopy solutions from large systems all the way to the miniaturized compact spectrometers. If you're looking for any laser at any wavelength, from the visible to the mid-infrared, any wavelength, Lars from nanoplus here with us to provide that laser. And finally, last but definitely not least, one of the success stories in Europe, Avantes, providing spectrometer solutions already for 20 years, all the way from the Netherlands. Thank you so much for supporting this event, and thank you so much, Dr. Elena Beletkaia, for your hard work on this meeting. What's going to happen today?
Thank you very much, Jose, and it's great to hear your enthusiasm. As I expected, I think it's doubled since you are out of your home office. But I hope that we will triple it through the meeting today, because we will start with Dr. Reddy's presentation. So from the end user who did prepare a lot of different challenges for the supply chain, we will have presentations of quite a few solutions. But the supply chain, you can see here who registered for this meeting. We have quite a few different end users. So of course, process monitoring is important in nearly every industry. And we have plenty of spectroscopy solutions. We also have a lot of system integrators and those who are providing the machines for process monitoring, so the full monitoring systems. But of course, we also need sensors, detectors, lasers. We need cameras, we need fibers, we need different components. We need software, we need photonic integrated circuits when we want to miniaturize all of this. We need microscopy. We also always need support from market intelligence and consultancy. And last but not least, we always need a support from R&D to keep innovating and keep going. So I don't want to steal any more time from our speakers and all the people in the room. Let's just get to this discussion.
This slide,
And
Elena,
find
only
out.
corresponds to the companies who registered for this meeting. So you're an Epic member. If you're an Epic member and you forgot, you see that you think that we forgot your company name, we didn't, we will never. It only means that you did not register for this meeting. Don't let it happen again. You have the calendar of online meetings, register as soon as possible for the companies that are relevant to you. And also let me remind everyone that you have to look your best because this meeting is live streamed in YouTube. So hello YouTubers of the world, thank you very much for joining our YouTube stream. Remember, if you have any question for any of the speakers, just write in the chat. If you want to get in touch with any of the participants today, all you have to do is drop us a line. I will make sure that we make the introduction. And this, of course, also valid for the people with us here in the Zoom room. Use the internal chat as much as possible. Use it, abuse it. Make sure that you talk to all the key companies that can actually do business with you. But if after the meeting you didn't get a chance to talk to the right company, all you have to do is send me an email and we'll make the introduction. Jose.pozo@epic-asoc.com. Elena, I want you to take the honor because you deserve it. Introduce the opening speaker. I know you are really almost as much looking forward as me. You're super excited. Thank you. It is an honor to have them. Tell us, who's opening the meeting today?
Oh, I will tell you more, Jose. Because I'm not at ECOC, I had a pleasure and an honor to have a short discussion with Atanu already before the meeting started. But now I will let all of you also enjoy the presentation from Atanu Sengupta, Head of Advanced Characterization Technologies at Dr. Reddy's Laboratories. Atanu, the attention of everybody goes to you. Let's share your screen, unmute your microphone, and see and hear you and your great presentation and all the challenges that you prepared for today. Atanu? We did practice today. Yes, so here we can hear you. Let's also share your screen.
Can you see the screen?
Not yet.
Not yet? Okay, just a minute.
Okay, now it is coming up, presentation mode, and perfect. And now all the attention is to you.
So hi, I am very glad and delighted to be here in this epic meeting discussion. I am Atanu. I head Advanced Characterization Technology Division in Dr. Reddy's. And a little bit introduction about myself. By training, I'm a spectroscopist. I did my PhD from University of Washington, Seattle, and in Raman spectroscopy. I was working in biosensor and diagnostics in the US for 6 years. Then I moved to Dr. Reddy's in 2013, and where Dr. Reddy's was kind of expanding and in terms of expanding their business to more complex generics and complex drug delivery platform. So in this presentation, in this talk, what I wish to convey is that I'll talk about my company. I'll talk about the capabilities and expertise we have. I'll talk about the product portfolio and therapeutic areas. And then slowly I will evolve more into the R&D and as well as the process monitoring, process analytical. And finally, I would like to spend a little bit time on my last slide where I'm going to talk about the wide spectrum of PAT technologies we are using in Dr. Reddy's and how and some of the potential going ahead, some of the unmet needs, some of the current challenges. I'll throw it out to you guys, and then I hope that will lead to some kind of food for thoughts for this for any kind of very insightful discussion going ahead in the session. And I can always connect offline. I can always take it outside the meeting. But yeah, this is my agenda. Coming back to Dr. Reddy's, those who are not aware of Dr. Reddy's is one of the leader in generics pharma. And in generics pharma, some of you are familiar, some of you are not. And it is little different from the innovator. While the innovator, you have the burden of discovering a molecule, going through the phase 1, phase 2, phase 3, which is time consuming, 10 to 15 years till we land to a kind of a right molecule that can be used for humans, right? But generics is a little bit a different scenario where we work with some off-patent molecules. The challenge is different. We need to take this molecule from scratch and make a formulation equivalent to the innovator brand. And while saying that, the formulation equivalent to the innovator brand, it is from scratch, because usually it takes 10 to 20 years the patent to go off out of the exclusivity. And the technologies that innovators are using, and we are using a more updated technology in terms of manufacturing, trying to compensate the time lag, trying to understand the innovator process, trying to come up with the right quantitative composition of the innovator and try to decode their design space and try to establish a kind of a product which is structurally and functionally and biologically, that means therapeutically equivalent to the innovator, it is itself a challenge. And if you look at the spread of the generics is in US, if you take the generics market, actually 90% of the prescription drugs are generics and 10% innovator. And you know the innovator, since it's a long-term R&D investment, the cost of innovator drug is huge. So in order to bring the drug and make it more affordable and reach to far corners of this world, the generics plays a critical role. So Dr. Reddy's is one of the top 10 generics company. Our annual revenue is around USD $2 to $3 billion. And we have different business. And in this presentation, I'm going to talk about the business unit where I am from. I am from small molecule and the small molecule global generic business. So we are catered to a diverse geography, and we have a strong presence in US, India, Russia, UK, and we are growing our presence in EU. So having said that, the company was founded in 1984, and then over the year, we developed a strong R&D capability and expanded our capability in diverse therapeutic areas. And here are the few of the platform technologies we have. Here are the few of the therapeutic areas we have medicines out there commercially. So now going to the next slide, if you don't have a strong R&D, if you don't have a strong infrastructure, this kind of diversity in portfolio is not possible. So we have platform technologies starting from liposomes, peptides, nanoparticles, emulsions, gels. It's a very diverse, complex portfolio. So our R&D has evolved over the last 10 or 20 years, focusing on 5 fundamental pillars. Definitely chemistry, because we are the leading supplier of API in India as well as globally. Then we are formulation. We got better and better in terms of making complex formulation. And recently there's a lot of focus in characterization, because slowly we are transforming into an API driven business to a complex generics driven business. So in a complex generics, the understanding of pharmaceutical material is very important. And for the pharmaceutical material understanding, you need to understand the molecule to drug product, and you need to characterize them from atomic level to crystal, to particles, to physical bulk property levels, from all structural hierarchies. And once you can do that, then you can understand the innovator, how they are made. Once you can do that, then you also decode the process design space of the innovator. So all these are very critical. So we have built a very high-end facility of advanced characterization. We have a state-of-the-art facility starting from microscopy, crystallography, thermal technique, spectrometry, mass spectrometry, high-end NMR. All techniques are there so that we can characterize the molecule from end to end. So, and this is where I work. And slowly and steadily, we are building capabilities in not only in finished formulation or the finished API. We are interested in the dynamic process attributes or the in-process attributes. And we are slowly and steadily developing capabilities in Dr. Reddy's in our manufacturing. And the next slide, will talk about more about our vision about process analytical technology. So as we all know that since the portfolios are becoming more complex and the regulatory agencies are pushing more and more to do quality integrated in design. So gone are the days where the quality is a stop, look, go. The quality is integrated in design, and if you integrate quality in design, and for autonomous process control, we understand very well that we need to follow and monitor in-process attributes starting from the synthesis and from the molecule and then the molecule formulated to a formulation, and then it has to be scaled up, right? And that is autonomous process control. But going beyond that, if you're seeing the disruption in the other industry, in the other manufacturing sector, and many of the innovator companies are started that journey. That is a journey from batch manufacturing step to continuous. And when you talk about continuous, it is more than controlling the process. It's much more how you can predict the failure mode, how you can recommend the corrective action. And not only the process analytical technology, how we come up with a kind of an integrated system, integrating the digital system, all the sensors in place, all the physical value system, all the instrument in space, and how you make a kind of a bot who can control your process, right? And if you, and the left side graph illustrates, it's just one of the analysis done few years back. Then if the generics companies focus in continuous manufacturing as well as automated process control, the amount of money that can bring, that we can save in the business. So it has a lot of implication in robust quality of the product. It is a lot of implication in how we reach speed to the market. And for generics, the business is based on speed. Okay, for faster we reach the market, we get a lot of market exclusivity. The longer we stay in the market, we just churn the business, right? So Dr. Reddy has a policy of first to file and last to exit. So we need something, we make market faster, and we also need to have a robust product or robust quality system so we can come back with the customers are trust us, right? So next, so we have made some significant efforts, and this slide kind of give you the whole landscape of the different processes, because we have APIs, we have different formulation from nanoparticles, microparticles, peptides, oral solid doses, or different functional release polymer. Just I wanted to summarize in one slide what are the processes we are looking at. We are looking at a reaction. There are ways how we monitor reaction. We are looking at weighting, how we weight a particle. We are looking at coating. We are looking at blending, sizing. And these are some of the techniques that we are working. Some of the techniques we have implemented in the manufacturing, in the commercial scale. Some of the techniques we are evaluating. Some of the techniques are out there, not in commercial as a commercial instrument, but we are working with academia, we are working with a partner to customize our instrument for us, right? So having said that, the last slide, I would focus on the different challenges what we face right now with the current state technologies, be it a spectroscopic tool, be it an imaging tool. There are 5 fundamental challenges in this journey. One is interfacing, because since we are generics, we have a diverse processes and diverse range of unit operations. So interfacing is very challenging in pharmaceutics, because there are challenges where the accessibility of the probe is not good enough. There are challenges where there is an aseptic process. We don't want to contaminate or we don't want to foul the probe. And there are challenges in the highly pressurized system, how the ruggedization of the probe, the instrument that we are working on. The second thing is sensitivity. For example, many of this formulation uses API, which is very low dose, right? Because when you go to a drug delivery platform, we don't need a lot of API. We can be specific to targeted delivery. So the API dosage significantly reduces. So now the challenge is, if I am doing a blending process, if I am doing some kind of a uniformity, a quantitative, if my signal, I am not getting a signal from the API, for example, I'm using a simple NIR or maybe a Raman. So where the detection limit around 0.1 to 1%, and the API is at 0.05%. So challenges are there. How to get more signal, how to get more resolution. For example, the formulation is a complicated matrix, right? There are other excipient we don't want. There are specific signals we want to monitor. How to deconvolute, how to go for a better resolution, because there is always a trade-off. If I understand that with the resolution comes with more optical element, right? And if you bring more optical elements, you are compromising the ruggedization of the instrument. It is not process rugged, right? So how do we do that? So similarly, speed of analysis. For example, we really want to move from at-line, offline mode of analysis than to real-time analysis. When you say real-time, I need to measure data at every minute, maybe every half a minute. And I'm dealing with a low-dose API, my measurement time is more than 5 minutes. So speed of analysis, having more data and having more quantitative data, having more spatial resolution, for example, coating thickness, how you overcome the spatial resolution, how do you see finer coating. In particle size, how do you have images to capture submicron particles? Because we cannot have electron microscope. Their technologies are not there to make it process friendly, right? So and finally, it's all about data. We cannot monitor process based on single instrument, based on single technique. There's a multiple integrated system, different sensors, different techniques, and we are collecting tons of data. And how to correlate the data, how the different technologies are harmonizing their data architecture, how easy for our data scientists and data engineers to extract the data and make a model, how robust, how the data analytics going, how the companies are investing in more in softwares, as well as the compliance of this data collection, traceability. So these are the few of the challenges. I am pretty sure that there will be more, but I will leave this up to the audience and then up to this panelist to take forward to further discussion. Again, thanks a lot. It is a pleasure to be here and the first time interacting with all you folks. Thank you.
Atanu, thank you so much. I think this challenge slide is something which we can keep for the whole meeting and just keep discussing point by point, going to everybody in the room and trying to find some solution for you. This is absolutely fantastic. Thank you so much. While everybody is digesting a little bit and coming up with the questions and comments, we did see that you are also in use of many different technologies. So what would you say is maybe one bigger global challenge? So why those solutions which you already have at hand are not sufficient yet? Or are those new challenges, or they are not accurate enough, or you need higher speed?
Yeah. So there are two things. One is technically and one is the culturally. For example, in terms of process monitoring, many of the other industry sectors have come up much more ahead than pharmaceutical. Pharmaceutical is a regulated industry. So, and recent only, I think 1 month back, maybe 2-3 weeks back, FDA has drafted a clear guidelines of the validation and the calibration and standardization of these technologies that are applicable to the pharma. So even the regulatory agencies are taking a little bit slow, because the definition, when we go to process monitoring and when we go to kind of a step towards continuous, there is no definitive batch size. So how we evaluate those technologies, how do you evaluate those models? How do you calibrate this? How do you evaluate the lifetime of any kind of model? Maybe let's example that I am making a simple blend uniformity model with NIR or Raman, right? Over a period of time, how I revalidate, what is the performance of laser over time, how we are doing the calibration, how that the model instrument change or some fiber optics change, right? So those are the challenges the regulatory agencies need clear clarity, and how when you make a method, when you make that implementation in the manufacturing in the pharma, we need to provide all the study and all these documents, and so that we know what are, so the method life cycle, model life cycle, that is one challenge. The second challenge is obviously the technology is becoming the manufacturing is becoming more and more complex. Okay, the thing is that the rugged instrument, for example, how rugged the instruments are. For example, today I was trying to talk to one of the vendors in Europe, okay, in implementing one NIR technology in our NFD. So the challenge is the NIR penetration is around 55 millimeter to 60 millimeter based on the vendors but now we have a working distance of 75 millimeter or 80 millimeter more. We have to access the material. Now the challenge is they are talking about fiber optics. Okay, so my question was, how the fiber optics hold if the blender is moving? How we ensure the ruggedization of fiber optics? So there are many things I can see. Yeah.
I suggest, thank you, sorry to interrupt, but since it already came up quite a few times about the fiber optics, and we have Viacheslav Artyushenko here from Photonics with us, and he actually had a question to you, and I think he also has some more to share with us, and maybe he can give you the answer to your question. So Slava, welcome to the meeting. Thanks for joining us, and tell us a little bit about Photonics and the question you had to Atanu.
Thanks a lot. Elena, I would like to say that it was excellent presentation, and for me it's great that, how to say, it almost completely coincides with philosophy which we try to develop, because what we would like to suggest, we would like to collaborate directly with Dr. Reddy Laboratory, and we have much more questions to discuss after the meeting, but here just to show very briefly, we already developed any kind of probe for any kind of spectroscopy which can be used in line. And we are not, how to say, trying to overload by big data computers, because everything must be done in real time. And data normally goes to cloud for real-time treatment of the data. But we see great advantages when not only one method is used, when it's a fusion of 2 or 3 methods, like fluorescence and infrared together, or near-infrared and Raman together. So that's why I'm fan not of the big data, I'm fan of the good data. So good data means, you should pre-select, you should filter what is really needed for your process. That's why sometimes it's enough to have only 2 wavelengths, to get good understanding what's going on for process monitoring. That's why, in addition to any kind of spectroscopy, which can be used for liquids, for gases, for granules, or blenders, just to remind that we have also at this event quantum cascade laser stuff, where it can be only few quantum cascade lasers selected to be fiber optic coupled, because every process needs kind of immersion. You should put probe in a reactor, in pipe, or in blender, something like that. So thanks for your for the chance, to show what we would like to suggest for collaboration with Dr. Reddy Lab.
Definitely, I would like to talk to you offline, definitely, because in some of the another technology we are evaluating is a high synergetic system, where we can do simultaneously the particle sizing, as well as we have a Raman attached to it, and we can see the crystal growth, right? So we are monitoring the crystallization. So definitely, if you can, the fluorescence is pretty interesting because it brings the sensitivity, and if there are some fluorophores are there which is normally not detectable by Raman and NIR, because of the sensitivity, we can pick it up by fluorescence, right? So
we measure spectroscopy, scattering as well, like detection of, let's say, few ppm water in kerosene. It's our current project right now. So that's why it's not only absorption of fluorescence, it's also scattering, which we measure to measure crystallization process or what else. So we are highly interested to collaborate.
Sure. Great. Okay. Great to talk to you. Yeah. Great.
Fantastic.
Sure. We'll connect.
Thank you.
For sure. We will make all the introductions and we will connect all the parties interested. But I would like to ask a question of my own, and sorry, Atanu, not actually to you, but to Doug from Tomra Foods. My question would be: we did hear here so many different challenges from Atanu in pharma industry. Is it something similar what you observe when you need to develop your systems for food industry, or the challenges are very different? And don't forget to unmute yourself.
Yeah. So thanks for the question. I think I really enjoyed the presentation. I actually did see a lot of parallels, right? So with what, particularly when you mentioned robustness. So when you talk about robust imaging systems or spectroscopy systems, that's very much close to our business, where we have high volumes of food in very adverse conditions, I guess. So we're installing spectroscopy systems in food processing lines, sorting hundreds of tons of product an hour. It's not always dry, it's not always clean. Yeah, that is something I think we share in common. Also, of course, resolution and sensitivity is something we always look for, though I would say the orders of magnitude are somewhat different. We don't need to go to the same level of precision as what I expect that you need in the pharmaceutical industry. I also was interested to see your comment about transferability of models and consistency of the measurement over time. That's something that we struggle with. Well, not struggle with, but it's close to our business, that we have to make sure that sorters in this part of the world are the same, giving the same sorting performance as South America and North America and China and wherever. So yeah, no, so it was very interesting. I don't know so much about the pharmaceutical industry, but I can see a lot of parallels in what we have to do.
Doug, it is indeed great to have you here. I am very excited to have two companies that are really looking at challenges. I'm going to go first to Anatu. Anatu, thank you. Thank you so much. I want three things to discuss with you. The first one, you showed as a challenge and as a line of R&D hyperspectral imaging. And together with my friend Thierry Emeraud from Lambda-X and many people in the room, we want to know what is your take on hyperspectral imaging? What is the problem you want to solve? And what are the challenges and how the EPIC members can help you on hyperspectral?
So thanks for this. That's a great question. And already we have capabilities of hyperspectral imaging. So our journey of hyperspectral imaging started with, the Raman imaging and the NIR imaging are hyperspectral. So we do a lot of chemical imaging. Mostly right now we are doing a qualitative imaging of a tablet, of understanding the distribution of the material, whether there are kind of different components distributed properly, how do you see the different layers in the structure and all those. And slowly and steadily, our expectation is how to make this hyperspectral imaging as a quantitative imaging. Can we estimate? Can you have a calibration-less estimate from the imaging data to predict the components? For example, I image a tablet. I have the drug, I have the excipient, let's say another polymer, and I see a nice image. But can I tell that how much of the drug, how much of the polymer, how robust is that image analytics? So, and also the spatial resolution. And when you are looking at the hyperspectral imaging, what are the kind of spatial resolution we are looking what is the state? So right now we are micron, because all the hyperspectral imaging mostly micron and submicron. So
and you're going to go submicron. So Thierry Emeraud from microscope and camera manufacturer Lambda-X in Belgium. Thierry, here is a challenge. We have hyperspectral imaging. They're already looking at that. They want to go beyond in the resolution. How can you help?
Yes, thank you very much to give me the opportunity. Yes, so can you see my screen?
Crystal clear. But if you go presentation mode, we see it even bigger.
Well, I try to activate it, but doesn't seem to work. Maybe
it's okay. We can see it. You are sharing with us your notes, so you go to display settings and you switch. It's maybe better.
So you don't have it full mode now?
We are seeing your notes. So we go to display settings.
Display settings, okay.
Here in front of you, next to show taskbar, top left corner. But yes, you know what? We can see it quite well, so it is okay.
So swap?
Yes, swap. Fantastic.
Okay, so that's exactly the point that Atanu was pointing out. Actually, we developed hyperspectral technology, which has a high spatial resolution compatible with microscopy, and also high spectral resolution, which make it usable for Raman to extract chemical composition using Raman spectroscopy. So you see here clearly an image of an API tablet. This is an application we've done with a university where you can extract everywhere on the image the chemical composition of different components, and then you can derive the distribution of the different API or polymers which will be there. So the way we done that, if I refer to your presentation, is not exactly online, but it is outline, as we need something like 100 seconds to be able to determine the full image, to solve the full image and extract the information, but which is quite already quite fast, meaning that you don't need to bring the sample to the lab on the standard, I would say, confocal Raman microscope, but you can do it in 100 seconds with basically a comparable resolution that does the standard Raman microscope. So that's actually a hyperspectral development at the beginning, but which turns to be usable for Raman imaging. So I would be very much interested to know if you are willing to use this kind of technology at line in your factory or in your process control setups or lines.
Can I ask you a question? Just a follow up. We can discuss this in more detail. I'd love to. So how user friendly is this? Because in a confocal system, what we use right now, it's a lot of expert driven measurement, right? So when you are putting in the at line manufacturing, is it based on some kind of simple to use and can be easily automated so that any user can use that, or it needs a skilled user to measure and analyze and interpret the data?
No, definitely, it's something that you will not use in the lab, but at line. So it's meant to be easy to use for let's say qualified operator to use it. But indeed, it's not a confocal, so it needs the sample to be put in a configuration because a confocal will just generate you a very local image. So it's a non-confocal system at the time, but if you spread or the sample on the microscope, you can extract the information. So it's really meant to be used, I would say, easy to use and compatible with a use in a production line, I would say, environment.
I would love it if a collaboration between Dr. Reddy's and Lambda-X started from an EPIC meeting, but this looks very, very promising. Atanu, one more thing. We saw all the challenges. Thank you for that slide. I always loved going to longer wavelengths. Mid-infrared is evolving, is here to stay. And I didn't see much on mid-infrared in your slides. Have you been evaluating that? And I would like to check that with Vigo system. Aneta is here with us. Chesh, Aneta, thank you for joining. Atano, is mid-infrared wavelength something of interest to you?
Exactly. Yeah, definitely. So we are also explore terahertz, far infrared. So beyond the far infrared. So mid infrared definitely interesting, but we have mid infrared spectrometer mainly in the lab. We haven't used it in the manufacturing because in some of the processes the infrared has certain challenges regarding moisture and ruggedization, because if it is a interferometry-based system, then how you make a ruggedized instrument. But in the lab, definitely we have mid-infrared.
Aneta, in the 3 to 12 wavelength range, Aneta, we always discuss whether there is a sensitivity for water constraints, and actually water doesn't absorb this wavelength range. If humidity changes in water concentration are a challenge for Dr. Reddy's laboratories, how can the mid-infrared wavelength detectors of Vigo can help here?
Yeah, thank you, Jose, for the question. It's a very good question. True, the humidity is disturbing with infrared detection, but there are some tricks or there are some wavelengths which are useful in this range. So I guess we can maybe further discuss this and find some solution. I would like to add also one more question. What do you mean by far infrared? Is it for you beyond 12 micrometers, or it's more after, let's say, 8 micrometers?
After that.
After that. Okay, so this is unfortunately beyond our scope. Let's say we produce photodetectors up to 16 micrometers. But still, this far infrared.
He's very interested in the mid-infrared wavelengths, Aneta. Don't look it back at that. He already does the ultra long wavelengths on the terahertz. I can tell you the mid-infrared is very interesting for Dr. Reddy's. Many companies in EPIC, Atanu, are doing the coatings that we have here. Iridium, the fiber. You already met Viacheslav Artyushenko, Viacheslav Artyushenko from Photonics, the detectors, the lasers, and we are very excited on talking to you. But I want to also talk to Doug Wright from Tom Rafud. I just talked about hyperspectral imaging. In the beginning, hyperspectral imaging was the trick in the sleeve to do the food processing chemical on site. How is hyperspectral imaging being considered in your process monitoring systems?
Yeah, it's a good question. So the answer is we use it already a lot. So we use both visible and near infrared, I guess in shortwave infrared hyperspectral imaging systems on a range of products, from nuts to French fries to potatoes to kiwis, blueberries. It's used everywhere. I think we don't generally go to mid-infrared wavelengths, as you were just discussing. As you say, the water absorption is too high. For our products, we do use it a little bit in polymer sorting, which is not in my division, but Tom sorts plastics. So that we use hyperspectral cameras there. One of the challenges we have is we really want to push the speeds of these systems and resolutions. We want not micrometer resolutions. Sub-millimeter is fine for us. But the product speeds are quite high, up to 4 or 5 meters per second of product. So it's a little bit, we are trying to do millions of measurements a second. So that's the challenge we have, is a speed resolution trade-off. And so we're always trying to push our imaging systems to that next level of resolution. And that comes with it also the processing power to crunch those spectra fast enough. So yeah, hopefully that answers your question.
It definitely does. And actually, EPIC member Voyage 81, which I think is in the room, Amir, if you're in the room, we have always a challenge of the data, the amount of data is generated in different processes like hyperspectral imaging, and reducing that data is actually the here. We're going to discuss this a bit later on, but now that we got the attention of everyone, with Dr. Reddy and Sonra here directing the discussion, I want to go to something that really struck all EPIC staff when we found out. Have you heard about holographic microscopy? If you haven't, be prepared to hear something very, very, very special. Yves Emery, thank you so much. Merci beaucoup for being with us today. We want to know what you bring to the table, and afterwards we have another round with our end users today to see how this can be useful and which other EPIC members can help you. The floor and the attention of everyone here in Bordeaux, the attention of everyone goes to you.
Okay, many thanks, Jose, many thanks, Elena, also for the organization. I'm very happy to present that. So yeah, I will show you how digital holography microscopy can help for process monitoring, both material and life science. Okay, I will have to run because we have, well, basically 6 minutes for the presentation. So if you have any further question or request for detail, you can still contact me after all, and we can get more discussion. So what is the real time for me, the process monitoring? I think this is a good example. This is wet chemical etching. And the idea is to know when you need to stop the etching. So you need to know that in real time. You don't want to make multiple experiments. So you need to be able to measure. This is measured in the liquid, in the etchant. This is measured through a glass window. There's an electrical field. We measure as it happens, and we can take decision. We should stop the process. We should continue the process. We can measure roughness, depth, and so on. And this is typically a good example of process monitoring. So what are the main challenges? Because we can say that, okay, we know the material, we know the etchant, we know that we need to take 5 minutes or 10 minutes, but reality is a bit more difficult. There is unavoidable variability and non-linearity of material and machine. If you think to a laser beam, well, it could fluctuate it to the change of temperature, or small misalignment, or whatever. If you want to make femto laser engraving, you have some stress in the material, so by refringence, and it may change things. So if you think also to some bio application, some cell will not react exactly the same, or well, many things can happen. So there are always small adjustments that are continuously necessary to ensure an optical quality, an optimal quality. So this is needed both for material and for machine. It's very difficult generally to take a sample out for control and to resume the process. Often processes take place in situ, in immersion, humidity, there is some gas, some temperature, some pressure. You need to, well, to make a vacuum, so you cannot take it out. So it's usually very difficult, and you are losing time in your process, so it's also costs. Well, you have usually a high throughput. It's expensive also to stop a process or to fail, and often, and so if something fails, you want to stop immediately or need to react immediately. So feedback should be immediate, and the process time scale can go from hours down to microseconds. Many samples go on flight. You cannot stop a chain. And sometimes you also have large areas, so you need to be able to screen very large area. So I'm here to speak about holography. I will not discuss your process, and I will try to tell you how DHM can answer many of these challenges for both life and material science. So I will start with material science and go back at the end of my presentation with life science. So what is a DHM in material science? It's a 3D optical profilometer with a so-called interferometric resolution, what means that vertically we are in the nanometer range, and laterally we are limited by diffraction, so we have typically micron. Generally, optical profilometers are rather slow because you need to scan either laterally or vertically or phase scanning to get your answer. DHM is immediate. So basically you don't need any scanning. So we can make time results measurement. We can make 4D, and you can read this is a graphene membrane deformation due to a differential pressure between the top and the bottom. Time scale is milliseconds. So standard system measure at 200 frames per second, but we also high-speed camera to measure up to 100,000 frames per second full frame. So we are very, very fast. Okay, how is it possible? As I said, 3D topography is measured instantaneously at camera rates. We do not necessitate any scanning mechanism for acquire the 3D information over the full field of view. So there are many advantages. I will discuss all of them separately with a few examples. First of all, there are no blur during exposure sensitive measurement. If a sample moves, accretion time goes down to even 1 microsecond, depending on the laser source we use. Acquisition 4D up to 100,000 frames per second, full film. And even if for MEMS we go to 25 megahertz, but maybe it's a bit less important for inline quality control. So we have many 4D applications from static to 25 megahertz. So what does it mean, no blur during exposure sensitive? So in environment, you often have moving sample, you have vibrations, because you get a vacuum flow, you get a lot of noise, a lot of people around. So we measure here, this is a laser hole, a soft hole actually with a depth of something like 800 nanometers. We can see we shake our system, the sample moves, you can see the glass of water, but we still measure perfectly the topography. So that's insensitive to vibration. On-flight measurement, we don't need to stop the sample to measure the 3D topography. Also, sometimes you have a lot of heat, it can be hot. So this is an example of a measurement of a hot plate, heating from 0 to 800 degrees in a few seconds. You get turbulences, but we can still measure it. And also you have some sample like this spiral spring. It's never static because it's floating. So it continuously vibrates. You need to measure it instantaneously. So it's, for me, we think it's a static measurement, but the sample itself is never static, it's suspended. So we need to measure it very quickly. Then what is for us 4D time measure, 4D real-time measurement? So we investigate response of sample to light irradiance, to chemical action, to a change of temperature, to a change of pressure. We have seen them. This is mechanical wear. This is a ball on disk vacuum tribometer that can also heat up to 800 degrees, and we measure in real time in each turn, so we can continually follow up the process and to see whether it has to be stopped when something happens. It can also apply to evaporation, to dissolution, to electromagnetic forces, or to whatever. So it's opened many, many new applications. It's also enabled to investigate large surface. It's an application for forensic application on paper. And well, optically, and the goal is to look at the pressure that the writer put on it, because with robot and everything, it's very easy now to mimic signature, but not for the pressure. So you have steep flank, it's rough. We need a 20x objective with a field of view of 300 times 300 nanometer, but you don't sign over 300 times 300 nanometer, but on centimeter square. So this image is a stitching of more than 3,000 images stitched in less than 1 minute. So we can also look at large sample to see what happened very quickly, and we can react. We can also perform fast quality control. This is roughness quality control on hip prosthesis. So basically, we have automated rotation. This is for both cap and ball for prosthesis, which have roughness over the full surface between 1 and 6 nanometer. And this is the case when we make on-flight quality of saw marks on wafer. The size of the wafer is 150 millimeter. The time of flight below the sensor is 0.7 seconds. In production, we have 1 wafer per second. We have 4 sensors, and we make the profile for each of them, and we make sure that there are no saw mark deeper than 10 micron. So that's very fast. It's also enabled to measure as you manufacture. So we have models that we can integrate into other machines. This is a polishing laser. There is a continuous scan of the sample, and we measure during the polishing. And we can see here, this will be what you will see on the screen in real time. So it's possible immediately to get a feedback to the laser, so you can adjust the geometry of the beam or the power of the beam of whatever, and you can make sure that you are really polishing it, but you are not deforming the surface. You can see here there is some overheating, and you end up to deform it. So this map, actually it's a topography map, so color encodes height with nanometric vertical resolution. So the DHM information here is exploited to control the laser beam parameter in real time. That's a similar application here as well for femtosecond laser engraving. So we have also a small model which is integrated here, and we measure the change of the phase induced by the femtosecond laser engraving exactly when we measure. So it's possible, sometimes depending on the bioinfinity of the material, of the beam parameters, we can really control and give the feedback, and then it can enhance the quality of the laser inscription of arbitrary phase pattern. This has been well demonstrated. So again, DHM information exploited to control the laser beam parameters. Another example is also where the DHM measure in real time some pattern produced by interference-led lithography, and again it's measured in real time, and the feedback is given by the laser. The DHM information is exploited to control in real time during the process the exact structure topography. So that's open many applications, can save time and money. So we also have, well, MEMS up to 25 megahertz. I will not spend time on it, but it's also a big part of activity to be able to measure up to 25 megahertz to characterize microsystems. Okay, so that was for material science, so we can really see processes in real time. So what is now a DHM in life science? So in life science, we work mainly with transmission system, and it's a label-free, so we don't need to stain cell to use dyes. It's very low light intensity, so we can measure over long term without disturbing cell. It's a totally label-free optical microscopy to observe cells in 2D culture and also 3D spheroid of organoids. We measure a phase of light which contain information on the thickness of the cell, which means the shape, the morphology of the cell, and the dry mass, which is related to the intracellular contents. So those are thickness and dry, two endogenous markers. So this is why I said we don't need any fluorescence dye, anything else. And with those two endogenous markers, we can, without label, investigate cell health status and dynamic bioprocesses. We spent 20 years to develop that, to try to understand, because we was the first one to be able to reconstruct phase and intensity from a single hologram without any scanning. So we worked a lot to interpret quantitative phase imaging in terms of underlying processes, and we get many patents. So in lab science, we have information of shape, of morphology, on cell health. We can see this one is dead. We can identify and quantify organelle. Well, this is typically liquid droplet at adipocyte that will be flash, that will be lost if you make a cell washing, because there is no adherence. So we have no marker, no cell washing, no cell preparation. And this is beating cardiomyocyte. So we measure here at 1,000 frames per second. The standard system is 190 frames per second, but we have also system measuring up to 100 kiloframe per second. So we can make dynamical recording. So the non-toxicity enabled to make long-term recording, and the speed of the measurement enabled to look at also milliseconds scale. We have demonstrated application for cell culture monitoring, for cell dynamics, we have Foley tracking of particle, sperm, bacteria, and also we have demonstrated optical patch clamp when we have many patents. So the workflow is very simple. You culture your cell as usual. You don't need any specific substrate, and cell staining, any pipetting or washing step. We can make acquisition at single or multi-site, from millisecond image capture at capture rate to multi-day time lapse. And we have software to interpret data, to investigate bioprocesses, and also for automation to make plots. So you can measure, you discover, you can publish, and also you can, without any issue, you can then also control the quality of your production, your process for life science applications. Well, you can see it's fully automated. This is a 384 well plate scan in less than 6 minutes, and we can investigate all the physiological aspect of cells simultaneously over the full well plate in that case, with long-term labs capacity. Okay, and this is an example where we were using testing the effect of doxorubicin on, well, I don't remember which cell it was. Well, doesn't matter, it goes for any type of cells. And we can also classify phenotypes. So its possibilities are really huge. So we offer a complete range of standalone system and also an add-on module. We can steal the reflection transmission system, the high-speed system. We have camera module that can be assembled on other microscope. Also, we have other modules that can be mounted on laser polishing machine. We have lenses system, industrial system. We have macro system for large area. We have option and accessories for motorized stage, environment control. So we really handle from sample handling to data analysis with also software for analysis. So we can measure 3D topography at in situ and analytical speed. It's enabled to discover new innovative research paths. You can perform quality control on production floor. And in the frame of this discussion and presentation here, we can monitor process in situ and in real time for both material and life sciences. Okay, that's it. Many thanks for your attention. And should you have any questions, do not hesitate to ask.
Thank you very much, Yves. And indeed, do not hesitate to put all your questions in the chat or raise your hand, and we will come and answer all of them. But before we get to that, as I said, it's my first meeting after the whole summer, so I cannot hold it much longer. I want to come to epic question. I want to challenge you, Yves, with an epic question to summarize very quickly what you can do for them, and maybe make a much more elaborate part of what they can do for you. So what this supply chain of photonics technologies all together in the room can do to help you further?
Okay, so we have quite a few collaboration with manufacturer, with other company where we supply modules that are integrated. We have the SDK software. So we are looking for collaboration because our goal is to extend the use of digital holography. So I'm sure there are companies who need such a system. So we are mainly looking for collaboration for people who has interest to integrate just a system in their system or to use directly the system as it is. And what you can bring, I think, everything I've shown about speed and so on is pretty unique. So I can bring them advantage for their product. It's also for pure quality control, enabled to have a fast diagnostic and to decrease cost and to save time. So this is what we can bring.
Fantastic. So if now we try to get back to those challenges that Atanu was bringing to us, and you also told yourself from your presentation, I mean, it was quite clear that you can do very well in 2D resolution. How about quantitative analysis? Is that something
it's 3D,
also
so
included?
Yeah, it's 3D. As I said, that was my third slide. I said we get an intermetric resolution. So we get vertically a nanometric resolution. So this is really quantitative. We compare height with a laser wavelength. It's not a scanning mechanism, so there is no drift. So it's a very purely methodological measurement with a very high repeatability in terms of accuracy. So quantitatively for topography, we go below the nanometer. And laterally, we get an optical resolution. So typically with a 20 or 50x, we are, well, 1 micrometer or down to about half a micrometer lateral resolution. So this is for material science. And for life science, it's a bit more difficult to quantify, to tell limits like this. But what we have shown is that in viability and so on, optical patch clamp, we get a resolution which really enable to give assessment and to quantify viability of cell very precisely. So with similar resolution than alternative technology when they exist.
Okay, thank you very much. Atanu, so do you already see how you can fit the Lyncée Tec technology into your laboratory or into your manufacturing line?
First of all, that was a wonderful presentation. Thank you.
Thank you.
So I see quite a few possibilities. For example, when we manufacture tablets and there are obvious quality defects. For example, the tablet surface are not smooth, the tablet, there is a hole or crack, and these are the usual case of a customer recall. They don't want some defective tablet. That is one area. The second area is that I was thinking of when you are talking about scales, right? You can have a kind of a very native state cell microscopy, right? And definitely I will pass this message to my biology colleagues in Dr. Reddy's. But from the small molecule, we develop a lot of microspheres. These are polymeric microsphere particles, and we are interested to see the internal structure, the internal porosity, okay, and how the level of drug encapsulation inside the microsphere. And those are kind of cell. They're the same dimensions in microns, and they are made of polymers, like lipids-like polymers. And did you ever attempt that before in terms of polymers and polymer delivery vehicles, kind of that?
Okay, so you had many questions. So I will try, I took some notes, I will try to answer them. So first of all, you speak about tablet presence or not. So I will say, I don't see where a vertical resolution is needed to see if a tablet is there or not. I mean, you can use a standard camera, you can still fill or not because a tablet is certainly a color. So typically, if you want to think how you can use a system, you should think, okay, the advantage is to have 3D information. So basically, and we have these sensitivities that you should use. So finding if a tablet is there or not, I think there are alternate and maybe easier traditional vision system that may do it, but maybe you have some idea of cases where it's difficult. So you should elaborate more so I can tell you if I can help.
The tablet, sorry to it's a tablet that not there was not my question. The question was if I have monitor the tablet roughness or tablet defects.
Yeah, okay.
So is it the profilometry we can easily do? And right now we are basically doing, and that can be a very nanometer size groups or defects during the coating.
So
and that can be done quantitatively, right? I assume that can be done.
Okay, so in that case, it's more rather than the present, it's more the surface topography of the tablet. In that case, yes, we could be able to do such measurement. We have to see which roughness, but if you tell me it's a nanometer range, this is definitely our area. The microsphere internal structure is interesting. We have some solution where we can even make some tomography by rotating the sensor. We can get many. So I guess those spheres should be, even if I'm immersed or whatever, they may be transparent. So we may have solutions. So as I said, we can make a lot of theory. The best is to test. So should you have any sample for tests, feel free to send it to us and we can see what we can do. Because we can spin for hours what are the difficulties possible or not, but we need to see the sample to efficiently give the right answer. Yes, we can or we cannot.
Thanks a lot. Thanks.
Okay, fantastic. We have more questions to you, Yves. And we have a question from Sergei from Optics. Sergei, do you want to unmute yourself and ask your question?
Yes, I just had a basic question about the DHM. So you talked about looking at while polishing the glass surface. I was curious if it's possible to do the same thing inside the glass, or it's limited to the surface monitor.
Well, the application I show with the femto laser is definitely inside of the glass because the structure, it was really engraved within the glass. So we have already did similar things when engraving fiber optics in glass or other things in glass. But the example I show with the femto laser, that's typically inside of the glass. Yes.
Okay, thank you.
Should I show it again?
Yes, please. Because I had the feeling it was just a surface polishing, but no, I was wrong then. Yes.
Okay, this application, this is really, sorry, I have to go back here. So this application is really within the glass.
Okay,
it's femto laser engraving. So I said that a change of the face or the color is really because that's something that we may have to investigate, but this is within the glass. So my answer is yes.
Okay, thank you.
I am quite amazed about what kind of things they can be done with digital holography, which is a topic that has been asked by many of our key companies in the segment. One of those key companies is one of our biggest EPIC members, Sony, and Faisal is with us representing Sony. Faisal, I think you enjoyed this presentation as much as I did. What's on your mind?
Hi, Jose. Thanks for the invitation. I am sorry I actually missed most of the presentation because I joined a bit late. I thought starting at 3. But maybe that will be a stupid question then because I missed the earlier part. But did I get it right that it's not only the surface monitoring, but you do the engraving part as well? So if you do the laser engraving in glass too, what's the optimization of the surfaces that you can do? Is it like a linear structure that you can build? Can you produce a three-dimensional curved surfaces as well? Or what's the limitation of the engraving that you can do?
Okay, well, yeah, I mean, there's many possibilities. So it's difficult to give a general answer because I'm not sure everything you have in mind. But yeah, we could also, because, well, digital is great because we can typically also make digital lenses. So let's say if the surface is really curved, we can compensate for this curvature digitally, and then we will still be able to measure without any problem. So, but okay, there are always some limitation. I'm not sure how curved it is, if it's a very small radius or not. So for me, it's difficult to give because I don't have enough specification. But okay, basically, we have reflection system. With the reflection system, we can measure interfaces. Well, we measure reflection interfaces, so it can be the reflection on the top layer, but it could be also reflection internally to the sample. So there are several possibilities. So we can also have an idea of make a kind of scan like this. We have transmission system as well, where we measure through the sample. So we'll integrate that, and then we will also see what happened inside. So yeah, we can make both. We may even think sometimes if it's very some pilot project, we could combine both. So yeah, there are both solutions, and we have this digital processing that we can make or we can correct for some curvature. We even make the digital. We can also make the focus digitally without moving mechanically the sample, which is also very great for. I had no time to speak about that, but typically when.
Sorry, would it be possible? We can take it offline. Could you just send me your presentation, and then we can carry on separately on that.
All the presentations are available. You will get all of them. There's also in YouTube, but you really have to speak to each other. Yves, you should know one thing. Just before the pandemic, Sony in Stuttgart asked us to organize a workshop only on digital holography. So imagine how important this topic is for them. But I want to continue this fantastic discussion. I want to bring somebody else to the table. We actually have a very important company in providing services to the Indian industry that wants to promote technologies from EPIC. Gaurav Shah, GAMP Services, welcome to the show. How can you help the companies in the room open up the Indian market, which is a key market in process monitoring?
Yeah, thank you, EPIC team, for giving this opportunity. So since I have seen this topic for process monitoring, we are also into the similar field, and we help the end users for providing a real-time monitoring products for pharmaceutical industries. So Dr. Atanu has covered the most of the topics and the processes which are mainly used in the pharmaceutical industry. One process which I would like to elaborate more, that is the high shear weight granulation, which is.
Can you repeat the process?
High shear weight granulation. It's RMG, rapid mixture granulator. So in that also we have the technology called direct force flow sensor. It's DFF sensor, which can help to monitor the process real time. So similarly, we are also open to collaborate with any other manufacturers who are really looking for the good distributors for India. So we are based at India, and we support all the manufacturers to connect with the end users.
Gaurav, be prepared to be introduced to many companies. This is going to be great. It was great to have you here. I'm going to Yves Emery now of Lyncée Tec. The first question to you is from myself, but I want my help, the help of Denconfocal to ask this question. I have to say privately, during your presentation, I went to Albert from Denconfocal, equipment supplier for automation of different solutions, and I asked him, can we integrate this digital holographic microscope into one automated machine? Albert,
yes,
that was my question. What is your answer to that?
Yeah, I think it might be possible, but yeah, so I think it's possible, but the main thing might be, and that's also one of my questions, is what about the required computing power? I'm not into detail. I'm not into too much details about holographic microscopy systems, but I can imagine that you need a lot of computing power, and if you want to do it in line in situ, so can you comment on that?
Okay, so I've shown some example. Typically with this solar wafer, when we process, basically we process 50 measurements, we stitch them together, and basically we get one wafer per second. So it's pretty fast, and we can, with a nowadays computer, okay, we use graphical card, but typically it's run on a normal computer.
Okay.
We have processes for bearings, for example, where that's ceramics bearings, and typically the idea is to make sure that there is no defect, because ceramics is very, very hard. So typically it rotates, and we need to measure the full surface, totally everything. And we need to make sure that there is nothing that can damage it. So basically we measure the full surface. It's again with a, I would say, normal computer with a good graphical card. So we process 12,000 images, and within, I don't know, a couple of minutes, it's done.
Yeah. Okay.
So now it's not a real limitation, I think.
You definitely need to have,
and I think the power of digital holography really rise with the increase of power of computer. It couldn't have happened 10, 20 years ago.
Yeah.
And maybe this is the reason why digital holography arrives a bit later than confocal lasers or other technology, because we really needed to have this computer power. But now it's here. So it's no longer a limitation.
Albert, you really need to have an offline discussion with Yves. This DHM is one of the most striking things we have seen lately in the EPIC network. Yves, another question to you, and this one comes all the way from the YouTube universe, comes all the way from Ireland. One of the success stories in photonic industry is the DFB lasers of Eblana Photonics, and they have a challenge for you. They're wondering if DHM is an adequate solution to monitor the quality and the mean diameter and topography of fiber optics.
We do have application on fiber optics, typically to measure the flatness of the issue. I mean, what is it, the quality of it? And we also, maybe also some solution more for R&D rather than to process also, but that's also a question of having a pilot project to have an idea of the structure of it.
That pilot project, we're going to make sure is set. I'm going to contact Eblana Photonics. We want this to happen. I know that I'm in the YouTube universe. I'm going back to our start today, Atanu. We have a question for you in the YouTube channel. And this question is about also from Eblana Photonics. This question is about laser-induced breakdown spectroscopy, LIBS. Is Dr. Reddy considering laser-induced breakdown spectroscopy, LIBS?
We already
destructive, and it will tell us. Are you considering it?
We already have one LIBS in our manufacturing operations. For example, what we do, we have particle characterization, the particle identification in the injection vials is a very critical quality step. So when you are getting extra particles that are undesired, so we need to know the source. All the investigation depends on what is the source of the particle, what is the particle composition. So we have LIBS, we work with LIBS, and so LIBS is pretty much there in our manufacturing. So good question. Yeah, LIBS is very important.
But LIBS is destructive. When I look at pharma and I think about the imaging processing, any destructive technique is a no. So how do you use LIBS?
No. So for example, if I'm doing a quality check of an injection vial, I find during visible check some particles. I want to know what are those particles, right? There are two ways to do that, either chemical by Raman or NIR, or for elemental SEME racks. But in our manufacturing, we brought an hyphenated LIBS and Raman system. So when you look at that extraneous foreign particle, we can tell you what is the elemental composition and what is the molecule. Then we can do the investigation. Where do we get this stress particle? How there is a containment challenge or there is a contamination challenge. This is that's how we use LIBS.
So how about you could have a miniaturized Raman solution that could be very, very close to these particles? Let me finish this. Dieter from Wasatch Photonics, how could you help giving a miniaturized Raman solution for taking away these destructive LIBS?
We can do that. Well, that would be great.
Well, miniaturized depends on how miniaturized you want it to be. So it's but you want to focus through your tiny particles. So it's a lot in the front optics, and then the Raman system. Then you want to have small and low cost, so you can actually use it in various places. There are ways to miniaturize Raman to something that's about the size of a computer mouse. And with everything integrated, so that is something that will be coming. The technology on the imaging front and on the sensor front has evolved a lot. So these particles you can then identify, like you said, with a tiny focus, with a tiny laser focus. You need to get close, right, to make the focus small. And I don't know where those particles are. If you have like this, like a deep beaker or something, or if you have them on the filter paper, it depends a little bit on that. But happy to talk to you about this offline.
You have to discuss this offline because there's a lot of secrecy here. But this Anatu is one of the challenges, and the next one. Anatu, pay attention to this. We're going to talk about terahertz now. And for that, we're going to go to my favorite city in this universe. We're going to go to Berlin. We're going to go to Fraunhofer HHI because of this collaboration with Fischer and Optica. They managed to do something spectacular on terahertz that I think Dr. Reddy is going to love. Lars, danke for being here. The floor and the attention of everyone goes to the coolest thing I heard lately. Goes to you, Lars.
Hi, thank you, Jose and Elena, for the nice introduction. So, we at Fraunhofer are basically doing applied research, and a specialty about the Fraunhofer HHI Institute in Berlin is telecommunications, and we're doing a lot of optical telecommunications. But there are some of us, like my colleagues and I, we're doing actually sensing based on this those components from telecom industry. And probably the biggest thing here is the terahertz sensing. And I will talk shortly about an industrial application of this terahertz sensing. And so the application, the application I'm talking about is the car industry, the measurement of the thickness of the car paint on the car body. So if you look at this car paint, it's a structure of multiple layers in the tens of micron range. And the task you actually get there is to measure the thickness of all the individual layers with a single measurement. And of course, this measurement has to be non-destructive, but also non-contact, since the paint is fresh and has to be stay shiny and so on. Also, of course, the process has to be possibly fully automated. And as you all know, that the car body composes of metal parts, but also plastic and some fiber enhanced structures. So it has to be fully independent of the substrate. And that's something at the moment only terahertz can do. And the process is the following. Actually, it's quite similar to what you might know from radar. So we send out, for example, a terahertz pulse, so electromagnetic pulse in the terahertz frequency range onto our layer structure. And then there will be a reflex, partial reflex at every interface between different layers. And with our detector, we can detect all those reflexes. And because the frequencies are that high, so the terahertz, we can actually distinguish the peaks from this, all those individual layers in the time of flight measurement. So from the distance of those peaks, we can then back calculate the thickness of the individual layers and thereby measure up to 5 layers simultaneously. The minimal thickness we could measure was 5 micron, and the accuracy is much below 1 micron or 1% of the thickness. And of course, since this measurement is fully in reflection mode, it does not matter what's below the below your car paint. So it could be metal or plastic or anything. And so this is kind of where the car industry is going now to measure the thickness of the paint layers. And that's, as Jose mentioned before, it was a project we did with, or a collaboration with Optical Photonics and Fisher. And one of the results was an ellipsometer-like measurement thing, which is shown here. That is, the idea was here to measure the chemical or the properties of the individual layers to have a characterization. But what we also did is, you've seen it on the first slide, and you can see it here again on the right-hand side. We attached the measurement head to a robot arm so that it is able to measure the car paint at several positions around the car, so that you have some key positions, and from that you have your reference in the car paint manufacturing. So our IP, or what we do actually, is we start with the semiconductor to generate the terahertz, and we also do like the sensor system around that with the optical driving unit and the data acquisition. And then what we are interested in is to find new partners, for ones which are interested in the terahertz technology, but also would like to include terahertz sensing in their portfolio, and of course system integrators, which help us, like Helmut Fischer in this case, help us to bring our sensor in the production line. And with that, I would like to, I'm open for your questions.
Thank you very much, Lars. That was really a great introduction to terahertz technology, and also a little shift to a different industry on where it can be applied. So I will try to pull you back into our pharma discussion over here.
Thank you.
Because as we're talking, you were talking about measurement of different layers, and at the same time, Atanu had a challenge about measuring the coating of the tablets of the pills. So is that something which you also could solve with your technology?
Possibly. Yeah, so the layer thickness measurement depends on the material contrast, which you have from the coating to the inner side. And this is basically what you need is a refractive index contrast of the one material to the other. And I could imagine if for example, if the tablet, the inner part is porous and the outer part is kind of a closed dielectric, it might be a quite good contrast. So I think it's something worth investigating further. And of course, the layer has to be on maybe a few tenths of micron or bigger, then it's could be rather easy, I would even say.
I see that Atanu is already ready to make a comment here.
Mm-hmm.
Yeah, so I think thanks for the presentation. Yes, we explored Terahertz technology a few years back, and we are evaluating that. Mm-hmm. It's a very good way of monitoring coating thickness, as you said, that different material, different refractive index, and we will do very non-invisibly coating thickness. And it's a very attractive proposition for the QC. And if you can automate the tablet measurement to hundreds of tablets, is much faster than SEM, much faster than other microscopic techniques, right? So there is a potential. Only challenge we used to have the cost, because the technology is very expensive, and frankly, the business is not keen of investing in scaling this across the different manufacturing. So, but it has a huge potential. Only one question I have. So do you see the resolution going down below 5 micron, and or what are the technological challenges that restricts to 5 micron? Because so
the resolution is below 5 micron, it's below 1 micron even, but the thinnest layer, which gives us enough signal, for that we would need to, like, if you're thinking of 4 micron, that's quite possible, but 1 micron thinnest layer, that would be a really big challenge. So we're going, of course, doing our efforts in this direction, but this would require a factor 5 higher frequencies. So a factor 5 higher bandwidth, which is not easily done. And it would might take several tens years maybe to go that high. But then you're starting to going into the range of the far infrared, right?
Yes.
And I believe your coatings might be transparent there as well. So this could be actually an interesting part there.
Okay. And
I would like to bring one more person to the table of this discussion on terahertz. We have Meng Pan with us from Lighted, and she just read my mind by posting her comment over here. Meng, would you like to unmute yourself and introduce Lighted to us, and also take your part into this discussion?
Thank you for your for introducing me. In fact, I just see the work that realized in the front force. That is amazing. During my PhD, in fact, I have also know some colleague who works in this problematic. It is great, and I think there are many potentials in industry. In fact, in our company, Lighted, we also provide the terahertz device, and in particular this year, we also provide the completed terahertz imaging solution. Different like our colleague, we works with the Terahertz source. So it is a source which I will say which more compact, and probably it is more cost effective. But we are suitable for the different problematic. Instead of the greater resolution, we can provide the better penetration. For example, we can check the the tablet or the leaflet inside the drug package. So if there is some needs, you're welcome to contact me, and I can provide more information. Thank you.
Thank you very much. And in the meantime, we also got a question from Sergei from Hydro Optics. And I think maybe both Lars and Ming can comment on the question. Sergei, do you want to unmute yourself and ask it?
Yes, thank you for the nice. Yes, you hear me? I hope so. Thanks for the nice presentation. I had a question related to some parameters of the laser of terahertz source that you use. So for example, the pulse duration, is there any restriction there in terms of power? And what sources are those? Is it through nonlinear process, you go down from near IR to terahertz, or is it something else? Thank you.
Okay, so what we're doing is optoelectronic terahertz. And that means that we have a photoconductor, so which is a semiconductor actually, with an applied voltage. And now if you shine light on it, for example, a short laser pulse, then this one gets conductive for a short period of time, and you get a current pulse, basically, which is then emitted into free space. We also do the same thing in continuous wave, which works actually similar. So in this case, it's not like classic nonlinear process, but this is photoconductive thing. And the laser pulse length, usually we work around 100 frames per second. That's the laser we use. Sometimes a bit below or a bit above. And the optical power is around 20 milliwatts. And the terahertz power, I think you were asking also for the terahertz power. We recently published a record of 600 something, 600, I think it was 37 microwatts of terahertz power.
Yes, great. And just a quick follow-up, the pulse duration, does it matter? Does it change the resolution, or?
The pulse duration translates into the optical pulse duration translates into the terahertz pulse duration, and that again is correlated to the bandwidth you get. And if you have a higher bandwidth, you can get higher resolution. So if you go for longer pulses, if you would go, then the resolution goes down, basically. But it's something for many applications would be also possible to have a lower resolution.
I was just thinking if it's possible to use, let's say, different technology lasers, like a semiconductor direct terahertz emitters, like QCLs, for example, quantum cascade lasers. But okay, this is why I asked those questions. Thank you.
Okay. Fantastic. Ming, do you maybe have something to comment on these questions as well?
Yeah, thank you. In fact, for our resolution, we use the different source as I just mentioned before. We use the millimeter wave source, so it's we works around the 150 gigahertz, but we also have the information about the phase. And with our dedicated software, we can also use to detect the thickness of the different sample. And in this way, even we work with lower frequency, we can achieve the resolution around 5 micrometer. So I think it depends on the application. We can provide the different solution.
We have so many things to comment, and the problem with this, I wish this meeting lasted for several hours, but we still have a lot of time. So let's continue with something that, for my opinion, is very, very interesting. Atanu, for Dr. Reddy, I hope you like short wave infrared technologies, because I'm going to tell you, I'm going to give you now the company that is making a big difference. Francois, all the way from France, new imaging technologies. Are you with us?
Yes. Yes.
Francois, we talk about so many different technologies so far. I want to go into the shorter wave. Tell us how you can help some of the challenges that Dr. Reddy presented before.
Hi, Jose. Hello, everybody. Thank you. Do you see my slide?
Not yet. It's coming all the way to Bordeaux. Not yet here. Let's see. Can you try to share again? Bottom of the Zoom window, you press the green screen up and then select, very important, select the screen that you want to share with the planet.
Is this any better?
Not yet, no. Bottom of the Zoom window, share a screen, and then select the screen. Sometimes people forget to select the screen.
Can I select the application, maybe? Oops.
Yes, you manage. You make sure great detectors and great sharing screen processes.
Thank you. Okay, do you see it in PowerPoint mode? It's fine. Okay. Well,
no,
a lot of talk.
You do need to switch the
switch?
screens,
okay.
so we would see it best. So just
There
one
it
more
is.
step in this long process, and there we go.
Okay, thanks. Well, thank you. We did have a lot of, covered a lot of subjects and a lot of different technologies. I just want to do a quick introduction of SWIR for those. We mentioned a lot of near-infrared and mid-wave and long-wave terahertz, and very quickly on shortwave infrared. Shortwave infrared is usually between 1 and 2.5 microns, and it's interesting the wavelength because you can see different things. Typically in semiconductors and solar panels, you can see through the silicon material and see the defects. So crack detection, dye inspection, or for alignment for semiconductors. And in solar panels, you can do either crack detection or functional inspection. So that's why this wavelength is interesting. Tamra and Doug mentioned waste and food sorting, and this is indeed interesting, both in hyperspectral imaging and also in humidity detection because of the absorption of the water at 1.4 micron. So that's why it's also a wavelength that is interesting for this. Of course, for photonics, there's also some interest, of course, because many lasers are within this waveband. And so whether you want to do fiber inspection, beam profiling, laser alignment, wavefront sensing, SWIR imaging sensors always answer these problematics. And then you have high temperature thermography. So with SWIR wavelengths, you measure temperatures above 300 degrees, but you can go from 300 degrees to 2000 degrees. So this gives quite a large temperature span, and it's used in hot glass inspection, but also in welding monitoring or in 3D additive material monitoring for metal. This is because you actually both can see the image, but also have thermal information of the cooling process. And so given the information on the cooling process, you could have detailed information on the cooling curve and thus be able to detect during the process any cracks that could appear due to this. Now, in a glance, in IT, we have a portfolio of sensors and cameras going from a QVGA high speed to a megapixel 1280 1024, a line scan, and with different interfaces for the cameras from GigaVision, SDI, Camera Link, USB 3. And at NIT, we do everything in-house. So we do the design of the readouts, the design of the in-gas PDA, the in-house hybridization, and all the development of the cameras and electronics. And so we cover the complete spectrum of what is necessary to offer the most dedicated products and with some unique and patented pixel designs within the pixel.
Thank you very much. I think we have to be there quite open. Atanu, there's lots of challenges to cover with all the wavelengths. I don't want you to comment on this one yet, because now in the open video, you also how excited I get about the next presentation. It's because BASF created its own company on near infrared. Atanu, I think you guess where I'm coming with this. I want another Trinamics. I want to give the floor now to one company that means a lot to Epic. It's one of the first success stories we came across in our network. We go to Wilfred Hermes. We go to Trinamics. We want to hear the current status. Most important, Wilfred, we also want to hear how the other Epic members can help you be even greater than you already asked. The floor and the attention of everyone goes to Trinamics, a BASF company.
Thanks, Jose. Can you hear me?
Loud and clear.
Slides you can see as well?
Perfectly clear. So
thank you for giving us the chance today to present about our technology. So first of all, I would like to introduce myself. My name is Wilfried. I'm a chemist by training, and since the beginning of Trinamics with the Trinamics. So let me introduce Trinamics. Trinamics, as Jose pointed out, is a fully owned subsidiary of BASF. So we belong 100% to BASF, and we were founded in 1st January 2015. We have, meanwhile, more than 200 patents, and we started at that time with 15 employees, and nowadays we are more than 170 employees worldwide. Most of the people are here located in Germany, in Ludwigshafen, close to our mother company, where we also have our production site, but we have an own site now in Shenzhen, in China. We have colleagues in business development running in US, in Japan, in Korea, as well as in China. Trinamics has two different technologies we're working on and two different product lines. So the first one is 3D sensing. So what are we doing with 3D sensing? 3D sensing, we developed a new way to measure distances and get out information. We started years ago with this, and all the way down now we are at the point where we can say that we set a new standard for biometrics. So what we developed is a measurement system with off-the-shelf components with our algorithm to ensure face unlock behind the OLED. So what we're doing is bringing the system behind the OLED screen and then ensure that your cell phone is unlocked in a very secure way. You can use this technology for other applications as well, for example, in automotive and robotics, but our main focus is here for the moment for consumer electronics, bringing our technology behind the display into smartphones. So where I would like to talk about today is about infrared sensing, as we call it, spectroscopy, and I have the pleasure to give a talk 2.5 years ago at EPIC meeting. It was at that time in Netherlands at Avantes site. And at that time, I presented to the community those chips I was very proud of that we were able to produce here in Germany in the semiconductor at selenide from 1 to 5 microns. And what I would like to show you today is what we made out of it. And first of all, thanks to the complete community, we got a lot of fruitful collaboration out of it. So when it comes to assembly, when it comes to dispersive element, when it comes to filters, to optics, all that stuff we cannot do alone. We need very strong partners, and that's what we did in the last 2.5 years. So let me start first of all with our spectroscopy business. So as you guys all know, we haven't invented spectroscopy. Spectroscopy is known on near spectroscopy since years. What we do and why are we doing this? We make spectroscopy accessible and affordable, and our vision is to bring near spectroscopy to the consumer. Today, I would like to focus on our B2B part, and we launched last year a full solution to the market, so the dynamic near spectroscopy solution, and the application areas where we're working on is circular economy. What we do here is on-site sorting of different plastic streams. The next one, which we are working on, is farm to fork here, especially in the area of feed and food. Then we go over to material substances, and last but not least, to body and hair. What we do in the last one is measuring, for example, skin analytics, the lipid and moisture level of your skin. And this is also the area where we move from the B2B version to the B2C. So here we are working heavily on miniaturizing further our spectroscopy to integrate it into smartphones. But let's keep for the moment at the B2B part. So we have 2 versions which we launched to the market. The first one is the reflection spectrometer, which I also have here in my hand. It works from 1.4 to 2.5 microns. It has 1% wavelength resolution and only immovable parts. The light source are light bulbs, and you can replace them. There is a battery power inside that you can do 6,000 measurements, and it works in, as mentioned before, reflection mode. We have a second version, which we also use at line or online, which works with cuvettes. Here you see a picture out of it in a BASF lab, so in our mother lab, where we measure close to the process at line samples, and we will launch later on this year the application together with our mother company. We will not, or we do not end with our spectrometer. So our solution is based on 3 pillars. The hardware, as pointed out, the spectrometer, but we also do the chemometric models. So we have comprehensive chemical know-how, since we are coming from one of the biggest chemical companies, to qualify and quantify analyzing materials. So we do the chemometric models on our own. So we develop them all here. Thanks to our mother company, we also can work very close with the big analytical lab of BASF. So we updated all the chemometric models to our device via cloud. That means that all our chemometric models are stored in the cloud. So the cloud is connected to software wireless, and you can use yeah, you can use the device anywhere and anytime, and the application is now running for iOS, for Android, and for Windows. So this is what we offer, our spectroscopy solution, including hardware, software, and the connection to the cloud, for mainly where we're working on the application on ourselves in the area of circular economy, farm to fork, material substances, body and hair. We have a second business where we are very active, and that's where we came from. So in our spectrometer is, as mentioned before, our own detector. And so we are producing the detector and selling it to different industries. So our IR detector runs in different applications. So for flame monitoring, it's used for spark and flame detection, gas sensing, moisture monitoring, and near spectroscopy. Near spectroscopy I mentioned before, but also other companies are using our detector to build up customized near spectrometer for online processing, offline processing, or benchtop spectrometers. So what are the unique features from our detector? So we can customize the pixel numbers from 1 to 256 pixels. So 256 pixels is our linear array. It's a very cost-efficient integration of spectroscopy capabilities into PUT. Easy and fast to adapt the hardware, and we have a broad wavelength range. We have two product lines running here. One is measuring from 1 to 3 micron, one is measuring from 1 to 5 micron, and we can also mix them up to deliver the full wavelength range. Its small size and the scalability is ideal for integration in industrial machines and appliances, and there are no solid, there is a solid state, this is a solid state version, and there are no moving parts involved. So this is what we bring to the table for the spectroscopy community, a handheld device, which is connected to every smartphone, to every app, and a chip, a sensor or detector, which can be used in different setups. So with this, I'm already at the end, and it's open for discussion.
Thank you very much for this introduction of this success story. And it's so great to hear that there are so many collaborations in place that you already have, and hopefully we will create some more over here. There is a question from Faisal from Sony. Faisal, what's on your mind? What do you want to ask from Trinamics?
Hi, thanks a lot for introducing your product range. I was just wondering on your spectroscopy range of the products. So you have from 1.5 to 4, or sorry, 1.5 to 2 or 2.5 micron range. So how many bands do you use within that? And what is the product application range? So are you looking at the lipids, ceramics, plastics, and do you actually change your wavelength range depending upon the product range that you want to apply your application in? Or do you have a more dynamic system in which the wavelengths specifically can be picked for the application that you're trying to run it on?
It's a good question. So for our spectrometer, we have 256 pixel array inside with a resolution of 1% between 1.4 and 2.5 microns. So all the different wavelengths are measured within this device. But we, in parallel, when it comes to dedicated application, we're also working on to remove the cost only for few wavelengths. So we call it multi-pixel solution. So for example, if you exactly know what you do, especially when it comes to process analytics, you always have the same process. Then you can, for example, maybe also work with 5, 6, 7, or 8 wavelengths, and this we can do customized. But for our handheld version, which we bring for different applications, it's so to say our generic product we run into with 256 bits.
Yeah, so that's what I was kind of worried about, the product application range. So if you have a product which actually has, let's say, 1 to 256 pixels, and you can tune each one for a different wavelength, then you can't. It's like taking a Ferrari and going to pick up your son from the school, which is like down the side, down the lane. So the application specifically, it wouldn't make sense to have such a costly product to run in a very specific application where you can run away with 2 or 3 LEDs, specifically targeting one application. This was my similar concern with the previous presentation, where the shot to SWIR range was presented with a solution with food and waste products, and then to the plastics and high dynamic thermal measurements as well. So the trend in the market is that you produce these high dynamic range solutions only for the R&D purposes, but when your application is specified, then you go to a lower cost solution which can be mass produced too. So what is your comment on that?
This is exactly what we're doing. So this is a cost point, let's say lower than 10,000 euro. So not all applications can be feasible for this device. And we are targeting going from the B2B version, really in the volumes, more to the B2B2C, in the end to the B2C, and there we really select wavelength which we need. We cannot go with our full power solution into this application. We really run there with less wavelength, but we know exactly where we're looking for. So for this application, we also don't go in a generic way that we say, okay, we make a spectrometer integrated to consumer electronics, which can cover all. No, we focus on dedicated applications.
Okay, Wilfred, we might have yet another collaboration here for you. From Slava from Art Photonics, he has a very interesting question to you, maybe to also extend your applications for the device. Slava, would you like to ask a question?
Yeah, a couple of things. First, I think you have a great product line already, and it's a bit limited only for the applications when you should really go inside reactor, inside pipe, inside any container, where the conditions can be, let's say, flame, high temperature, toxic, what else, So then fiber optics will withstand these applications, and when they will be coupled with your devices, then it will be simply extended applications, because we work with BSF, and sometimes they have a lot of applications which are, how to say, non-friendly. And one more point, which is just same like Faisal asked, we recently made one prototype when we replaced near-infrared spectrometer, which costs promotion optics about 15,000. We replaced it by 4 LED just to measure moisture only in powder, in starch, in, let's say, sugar, flour. So that's why chances to reduce costs of components for 5 times when you will replace spectrometer for few LED, in our case it was 4, exists. And it will be a sensor which is with individual IP, which you can really read through the cloud, like you suggested. Great idea. So you can really measure, let's say, how sugar is dried in Cuba, sitting here, in Europe. So because it will be real-time measurements. So that's why there's 2 points. Why not to use fiber optics? Why not to replace spectrometer for few LED for dedicated applications?
If I can comment on both points. The first one, we are looking for partners for fiber optics. So what we're not doing is to integrate into the process. So there, I think other companies have more knowledge and know-how. So we are looking for partners to do so. So as you pointed out, high tox, chemicals, and what else, not friendly environments, there we need partners. The second one, you're completely right. You can reduce the cost if you go for, let's say, 4 LEDs and 1 detector, but you can also turn it around, take 1 light bulb and 4 pixels. So both ways are possible to reduce the price, and we are working on it. But for this spectrometer, what the aim was here is to have a generic handheld version, a mobile version, which is much more cost efficient than a benchtop version and cover different applications.
Okay. But we are highly interested, to collaborate in both areas. So if it will be fiber optic coupling for customer applications, and if you would like to reduce, let's say, cost of your solutions, for distinct applications, because there are a lot of processes when you should control only a couple of parameters. You don't need a broad spectrum.
Would love to do so. How can I get your contact info?
Well, we can put you in contact. That's exactly what we're here for. We will for sure introduce you to each other. I want here to introduce you, unless you're already working together, but either way, then to the audience, I want to introduce Lars from nanoplus. I know that it's your new nickname, so let's get to it. And tell us a little bit about the nanoplus sources, and let's hear from Wilfred. Maybe that's something what they're also looking for.
Yes, thank you. I hope you can see me and hear me.
We can see, yes, very well, and we can also see your slides.
Yeah, that's this slide here. So I like the talk a lot from Wilfried, and also the comment from Faisal is giving today and also in other occasions. It's like solving this chicken and egg problem we have in this kind of still new technology. So everybody's telling me, okay, the laser has to be cheap, and then we make the application. And I'm always telling, if there's an application, then the laser will be cheap, because it could be a very, very low cost product at the end of the day if we come to volume. So this is exactly what nanoplus is trying to do since 20 years, and we took quite few steps there. Producing light sources which are highly specialized and still be cost effective, right? So we're doing DFB lasers here between 760 nanometer and 14 micrometer. That would be for a very specific application for Trinamics and other customers. But we can also do Fabry-Perot lasers at these wavelength ranges when you need more power, right? We can do SLDs in these wavelength ranges. At the moment, we're only concentrating at 760 to 2.9 micrometers with quite good success there. And this exactly goes into these high volume applications that we are looking for all the time. And then newly the MER mid-infrared LEDs, which we can get all the way from 2.8 to 6.5 microns, right? So here you have top emitter with a very broad band. So having a very small band all the way to very broadband LEDs, this is where we are producing light sources and see ourselves as a vendor for these. So I hope, Wilfried, you're considering our cooperation soon. Thanks a lot, Elena.
Thank you very much, Lars. So Wilfried.
Also here, would love to get more insights. Elena, could you make a connection to us as well? That I can pick
Absolutely.
It up. In the end, for us, the light source is a big topic. So our spectrometer nowadays works with light bulbs, and we are working on different settings now with LED, with lasers. So would be very interesting to go into more detail here. In the end, for us, of course, it's about costs. But let's have a discussion how we can, or if there is a fit, and how we can come together.
Okay,
yeah,
fantastic.
Very nice, thanks.
Good. So we will make the introductions. Also, after the meeting, we can stay longer to have some more offline discussion when it will be already not on YouTube. But now let's move on to our final discussion, final presentation of the meeting. And we discussed a lot about pharma, we discussed a lot about food industry, even automotive. Now let's shift yet again to a different industry. And for that, we have Sergis from Hydro Optics, from Alps Lasers, in fact, with us, presenting Hydro Optics and their spectroscopy for process monitoring in oil and gas industry. Sergis, the floor, attention of everybody goes to you. We already see your slide very clearly, so go ahead.
Yes. Hi, everyone. I hope you hear me. So I'm Sergis Hakovian. I'm the project coordinator of the project Hydro Optics, and also I'm an R&D laser engineer in the company Alps Lasers that develops quantum cascade lasers anywhere from 4 to 12 micron and even above. So today I will be talking about our project, the Hydro Optics. So Hydro Optics is a European project that aims to advance in photonic sensor technology in near IR and also in the mid IR, range, electromagnetic spectrum range by using coherent light sources, in our case, quantum cascade lasers and laser diodes. So it aims to develop high accuracy analyzer for automated online measurement of residual oil and particle content in the process water in the oil industry. And also we are trying to combine online data with the process simulation for optimizing the processes in the oil industry. So here I'm explaining in few words what are our problems, what are we trying to tackle, and what are the solutions that we propose. So the first is the residual water quality in the oil refineries. So as we all know, when the oil is being extracted from the ground, it contains not only oil, but also many other constituents and also a significant amount of water. And after multiple recycling of this water, the water is being released to environment. Hence, the control of the quality of the water is very important. That's the first problem. The second one is, of course, the oil extraction efficiency. So with our device, we try to increase actually the efficiency. And the solution we suggest for this is to introduce novel type of a sensor that will have highly sensitive online sensors for the oil and water detection. It will also have hydrocarbon aromatic detection other than oil. It will integrate also novel type dual-com spectrometers for monitoring some complicated chemical processes happening in the process waters. And also it will have a possibility to do a continuous monitoring of oil and water, which is quite essential for our application. And as a last part, it will have also hyperspectral imaging module for particle analysis, whether liquid or hard particles. So this is a bit of the concept of our sensor. So we will be taking the we will be integrating already existing devices of our partners, namely Quantared and IRSweep. So Quantared has already a device for measuring oil in water, and IRSweep they do dual-com spectrometers in the mid-IR range. So our plan is to integrate this, to integrate also the novel developed devices within the project, namely hyperspectral imaging module and particle manipulation with ultrasound waves. And also integrate our novel liquid preparation module, which will allow us to do continuous monitoring of oil and water. And all this will be fit into a 19-inch rack device that is portable and will be tested in fields, in our partners' fields in OMV in Austria and Tüpraş in Turkey. So this prototype will meet the requirement of the industrial environment. It will have a sample preconditioning integrated with it. It will have optical mid-IR sensing devices for a continuous monitoring of oil and water, and also particle classification possibility. So as we decided to discuss here about just few technologies that we use in our prototype. So first is the dual DFB laser source. So usually for oil and water detection, what we do is we use a single color laser to make an absorption spectroscopy. But the problem there is that over time, the background noise keeps changing, and it is becoming difficult to assess what is the real quantity of oil in the processed water. So in order to overcome this problem and to be able to make continuous monitoring, we use two color laser sources, namely distributed feedback quantum cascade lasers, both in 7 to 8 micron region, to monitor at the same time the background and also the signal. And by subtracting, we can effectively get the concentration properly. This is one module. The second module is liquid-liquid separator. So in order to do the absorption spectroscopy, we have to separate the liquid phase from the oil phase. And this is done by, thanks to the development of our partners in Elena. This is done by a novel type of liquid-liquid separator, which is patented, and it is based on the centrifugal separator technology. And thanks to this separation, we could effectively do the continuous monitoring. And this is just a lab experimental proof that we managed to have the separation that is required for measuring the oil in water. And the last module is the hyperspectral imaging module. So the task of this module is to determine the size distribution and type of solid particles in the process water, and also to determine the size distribution of oil droplets in the process water. So for the first task, we use a hyperspectral imaging system with near IR, visible to near IR wavelength range light sources. And this is what you see here, that system, the HSI module. And here is real data taken from the real process water at OMV, actually, for our partners. So you here see a histogram of particle size distribution. And this is the RGB color of calculated image color of hyperspectral data of the process water, where you can see different colors with different hard particles with different chemical composition. And then the second, the second goal, the size distribution of oil droplets is done with a fluorescence imaging, where we use a UV light to excite the oil droplets, and we do imaging of it. And this is again experimental measurement of oil size distribution. And also this is a segmented fluorescence signals from the process water where you can see the oil droplets. So, using this image, we could effectively make predictions on the oil concentration of the process water. So what are we looking on today is actually we are looking for interest, we are looking for raise awareness about our project, and we are looking for possible applications not only in oil industry but also in different industries. Like we have been hearing today a lot of application of spectroscopy, and we believe that our prototype is capable of doing spectroscopy in many applications where you need an absorption line from 4 to 12 micron, because our lasers can be easily exchangeable. And if there's a need to be a study on a particle composition of various liquid solutions, the device will be capable to do this as well. And I may also add that for the pharmaceutical industry, it would be maybe possible to use, for example, we have very efficient lasers, frequency comms, and also DFPs at 6 micron, which matches the amide band. And we believe that there, for some protein analysis, it could be also interesting to envisage our prototype. So with this, I would like to thank you for your attention, and happy for.
And I would like to thank you for your attention. Not only you, I would like to thank everyone for this fantastic meeting. We are almost by the end, but I wanted to go once again to the rock star today. I want to go to Atanu, Dr. Reddy's Laboratories. In Europe, with the European Commission, we get fantastic research, and we need to test it. We need to test it at the location of key end users. Dr. Reddy's Laboratories is a location where we could bring these demonstrators for a test. Can you make me happier than you already made me today? Atanu, I think we lost your sound.
Yeah. Can you repeat that?
We want to bring this demo that we just showed from 400 nanometers to 10 microns. We want to bring it to Dr. Reddy's Laboratories for a test.
Sure. You are welcome.
Sargis, you and me are going to India. Congratulations. And I want to say to everyone that we are about to close the meeting, but we stay for the private session. But before I close the meeting, I would like to let everyone know that all the slides presented today are going to be available, and you all have access to the full slide and the full video. But I always like to say, what has been the highlight of the meeting? And the highlight of the meeting is that we had this key set of challenges, for the entire industry. If you can help with any of these set of challenges, jose.pozo@epic-aso.com. Drop me a line and we'll make the right introduction to Atanu Sengupta, because we want collaborations to start with Dr. Reddy. It has been truly fantastic. Until the very next time, which is on Monday at 3 o'clock, every Monday, 3 o'clock, on online technology meeting. Thank you so much.
Thank you.
Thank you.
Thank you.
So your video was starting without the sound, so let's bring the sound in the room.
All right, thank you very much for that. But I would like to say that the video is not important. What's important is the presence of the EPIC members. So let's close the YouTube now. Thank you, YouTubers. The meeting becomes private now. This is a close group of friends. You can ask all your questions.